So when you look at what we are looking, when you look at mitosis and cytokinesis, within the cell cycle, as we are going to see, cytokinesis differs. Cytokinesis, the way you can hear it, it is cytokinesis. So cyto is standing for cytoplasm and kinesis is moving the cytoplasm. When the cells have matured and they are towards, they are supposed to, they mature, and then when they mature they have to separate the cytoplasm. So that separation of cytoplasm to form two different cells, that is what we call cytokinesis. So in animal cells, it just pinches it, after it forms a cleavage furrow, as we shall see, and then it pinches off. There is a very big difference between mitosis and the cytokinesis. Mitosis is a process that precedes cytokinesis. You will come across a number of things and you will be told that there is mitosis sometimes. Okay, mitosis comes before cytokinesis. So cytokinesis is like the terminal end or the terminal goal of after meiosis. Mitosis is done, then cytokinesis comes thereafter. So in plant cells, they usually form a cell plate, as we shall see. So both of them produce two identical cells, genetically identical daughter cells. So now when you look at mitosis, as I told you, in the cell cycle, before you go ahead and form other new cells, you need to remember that usually you need to prepare for the cells. You need to prepare for the process of division. Otherwise, if the cells do not prepare for the process of division, you will end up with cells which daughter cells which are not identical to the parent cells. So usually there is what we call preparation, and during preparation we are going to see a number of things in preparation. So it is interphase. So that first stage before mitosis, most times people confuse mitosis with interphase. Within the cell cycle, as we are going to see, we have a number of items within therein. You have interphase, and that is the preparation stage. This preparation stage can take very many hours. As we are going to see, I'm going to break it down into different segments. The interphase. Now, after interphase, we have what we call mitosis, and after mitosis, we have cytokinesis. So the cell cycle is probably made up of three stages, I would say. That is to say, interphase, which involves preparation, and then mitosis, where we usually see actual separation of the nucleic materials, then cytokinesis, separation of the cytoplasm. So as we shall see, in interphase the cells prepare. How do they prepare? By forming other copies of the genetic material, that is said DNA. So basically this is simply an illustration still showing you how the process of cytokinesis takes place in plant cells. They form a cell wall in between as a result of vesicles gathering together. So I will not put a lot of attention to that. Usually the rates at which the cells divide usually varies according to the type of cell. As I told you earlier, we have a number of different kinds of cells, ranging from when you look at the cells which are found, the nerve cells. These nerve cells, the ones that conduct electrical impulses to the via your whole body, they divide once and they stop completely. They don't divide again. And yet, when you look at other cells which are found, let's look at an example. When you look at the rate of cell division in the skin cells, it is 2 weeks. Every 2 weeks they divide. But when you look at the muscle cells, if you compare them with the muscle cells for them, it is 16 years. That means they are almost permanent. They do not so much divide. When you look at the intestines, you can see that after every 4 to 5 days, they are replenished. After every 4 to 5 days, they are replenished. The same with the liver cells. So you can see now the difference there. So you can see that the skin is always exposed to abrasion. So since it is exposed to abrasion, usually it is the one that undergoes a lot of replacement. So it has the highest turnover rate. So cells in the skin usually take a very short period of time. Some cells are unlikely to divide, and usually Those cells which are unlikely to divide after they have been formed after cytokinesis, they march into a certain stage known as the gap zero phase or the G0 phase, and that is a phase of completely no cell division. Such cells, I can give an example, is the neurons. The neurons are the cells that usually after they have divided, they don't divide anymore. That is why it is very, very hard for someone who has lost any of these neurons. In case, for instance, some diseases like poliomyelitis affect the central nervous system, and then they eat away the neurons. Some people get trauma, like accident, and then the neurons are cut. Those ones, the neuron will never go ahead and be to form dividing to form another one. No, that one most of the times through physiotherapy they are just helped so that their muscles do not reduce in size, but instead the muscles retain function, but the neurons that were innervating them, they are no longer functional. So what we usually talk about here, we can also add on, when you look at the cells are usually divided into basically a number of types, different kinds, but for us, the ones we are going to consider when it comes to the cell cycle, we are going to consider the autosomal cells, the non-sex cells, the cells which are found in your small intestines, the cells which are found all over your body, except in the gonads. Those are the ones that we are going to consider. Now, when you look at the diploid cells, so there are ones they call diploid, and they're usually represented with the arrow with 2n. You can see 2n. 2n, this 2n, for instance, we all know that usually a cell is made up of 46 chromosomes. And of those 46 chromosomes, we usually say 23 pairs. So 23 is the n, and 2 is the number of pairs. So since there are 23 pairs, they are usually in twos. That's when we say 2n. A haploid cell, contrary to that, a haploid cell is usually n, meaning that it is having half the number of chromosomes. We usually inherit half from the mother and half from the father. So the n comes from the mother and the n comes from the father. They unite during fertilization to form a zygote, which is diploid. I hope we are at par with that up to there. Are we at par?
Yep.
Okay. So now when you look at the somatic cells, I've talked about them. They usually have about 22 pairs of regular chromosomes. And then after those 22 pairs of regular chromosomes, then we also have an addition of those two. The 23rd pair is the sex chromosome, which is not usually the same. Like in heterogametic sex, like the males. Like males usually are called heterogametic sex because they usually have one X and one Y. Homogametic sex, that is the females. For them, they have the X and X. So going, okay, the karyotype and so on. Okay, maybe, yeah, in this slide there's not much to talk about here. Okay, so I've talked about the haploid, I've talked about the meaning of diploid. Okay, when you look at the cell cycle, usually I told you earlier that it is made up of 1 interphase, 2 mitosis, 3 cytokinesis. After cytokinesis, what happens thereafter depends on the type of cells. There are some cells which are permanently in G0 phase, like the nerve cells. There are some cells which are which continuously divide, like the quiescent cells. Then we have those permanent cells which usually divide only when there is need, like the hepatocytes. So now when you look at the different phases in the cell cycle, it is continuous. Cells do not okay, when you look at the phases, we usually have the interphase is not usually just interphase, but it is divided into different categories, different stages, each of it taking its own time. When you look at interphase, as you can read here, it occupies most of the cell cycle, and sometimes it is known as the resting phase. This is because there is no cell division taking place, but this is the most active time when the cells is dividing. It is misleading to call it the resting stage. They use it the word resting phase simply because there is no space, there is no much division taking place, but in actual sense there is a lot of division. There is a lot of metabolic activities happening within. So it is divided into different phases. That is to say, the first growth phase, or what they call the G1 phase. That word G1 does not mean growth 1, it means gap 1 phase. So during the gap 1 phase, we usually see the cell organelles undergoing duplication. Remember, we are going to form two identical cells. So since we are going to form two identical cells, that means that we need to also have, if we had two mitochondria in the previous cell, the parent cell, we need to form two more mitochondria to form 4. So that 2 can go into one of the daughter cells, then 2 go into the other daughter cell. If we had only 2 centrioles, we need to form another pair so that we can distribute 2 in one of the daughter cells, then another in the other daughter cells. So usually what happens here, we see proteins, usually proteins being synthesized within this G1 phase. We see cell organelles undergoing lubrication within this G1 phase, and that is all in G1 phase. Cell organelles duplicate with each other, then also just cell organelles, and then proteins undergo metabolism of proteins so that we can break down proteins and generate energy, even a generation of more energy that is going to drive this whole cell through the cell cycle. Now, when you go ahead and look at the second phase, this one, this G1 phase or gap 1 phase, it ranges from 8 hours. The minimum is 8 hours. From about 8 hours, okay, it can go even, not the minimum, but it can go from 8 hours to years and years. Some cells spend years within that cell cycle. So then it is the longest. Then it is followed by the synthesis phase, which takes about 2 hours. In the synthesis phase, we usually see duplication of genetic material, that is to say DNA. When you look at this chromosome on this side here, when you look at this chromosome, you can clearly see that the chromosome is made up of 2 chromatids. These things are called sister chromatids, and they are held together at an area known as the centromere. The centromere is a point of conjunction or a point of junction between 2 sister chromatids. What happens in the synthesis phase is this centromere, usually this chromatid, it was originally one chromosome but supercoiled. So what happens is this chromatid divides and forms another chromatid, and there are 2, but they remain connected to each other. Originally you had 1, but now through the synthesis phase you have now 2, but they are still held together at the centromere. Then after that is done, that is the only event that takes place during the synthesis phase. Then we go to the next phase, which is known as the next phase is known as the second growth phase or G2 phase. It is in this stage where we now see we usually see a number of things happening. We see the cell now increasing in size, the cytoplasm of the cell increasing in size, the cell organelles is starting to grow, and then we start seeing energy stores being increased. So that G2 phase is marked with increase in size. Number increases in G1 and S. In S, we only see DNA being duplicated. In G1, we see cell organelles being duplicated. In the G2 phase, we see more energy stores being assembled. And that is all about the cell cycle. That is all about interphase. So it is misleading, a bit misleading for you to say that during interphase, the cell is resting. Actually, a cell is not resting, but it is preparing for division. So you can clearly see from this graph here, you can clearly see this is a graphical representation of talking about the process of cell division and how the mass of DNA varies with the mass of the cell. can you go ahead and explain to me why do we see that okay, can you go ahead and explain to me the mass, the variation in the mass of the DNA, if you don't mind?
The mass of DNA, like on the second graph or the first one?
The second graph is the one that talks about the mass of DNA.
Initially, that's from 0 to around 4 hours. It's constant and then increases, remains relatively constant. It falls, constant, increases, constant, falls, then constant.
Okay, now I want you to go ahead and give some of the reasons as to why we are seeing a constant rapid increase, constant rapid decrease.
A constant, it's during interphase. Is it the G1?
Yes.
Then for the increase, in the G2 there is DNA replication.
The increase is in the S phase. So if you are told to identify the S phase is marked by this rapid increase in the DNA mass. That is S. That is where DNA replication takes place, in the S phase. And the mass of the DNA remains almost the same throughout the rest of the G2 phase. It is the mass of the cell organelles and the proteins that remain, which changes. That is why when you look at the graph above that talks about the mass of the entire cell, it keeps on increasing. And then nuclear division happens at the 12th day, and you can at 12th hour, and you can clearly see that explains why both the mass of DNA in each of the cells and the overall mass of the cell had to decrease back to 2. And are you seeing that? Are you there?
Yes.
Very good. So um okay, allow me briefly just talk about something small here. Okay, when you look at DNA, how DNA is organized, DNA is organized in a way that one DNA molecule is usually wound around proteins. There are some structures known as protein histones. These histones here, they are the ones that histones are positively charged proteins. So DNA is known to be negatively charged. Did you know about that? DNA is negatively charged.
To be honest now.
DNA is negatively charged simply because of the possession of the phosphate group. So it is negatively charged. So the proton, the histones onto which DNA is usually wound, they are positively charged. So they tightly hold it together. So that means that now we can have this DNA being compacted within a very small space. So a combination of histone and DNA wound around it, that is what we call a nucleosome. And then we see more folding taking place until we form a fully fledged chromosome. Okay, so I think now you are okay with that. So that is the detailed structure, and I would like to talk about telomeres and their significance. Say, when you look at the DNA, I believe you see these telomeres and their significance. Telomeres are very important, especially during this time, during this era when people do not want any more of aging. These telomeres, for your own information, they are the ones which are behind aging of cells. Cells are able to age simply because their chromosomes have telomeres. Telos is the Greek word from which telomere was obtained. Telos means a tail or the goal. So they are nucleotide sequences which do not code for anything at the end of the chromosome. And as the cell goes into the cycle, the more cycles that it goes through, the cell cycles, the shorter the telomeres. And as the telomeres keep on shortening, it reaches a point when the telomeres are too short, the cell undergoes what they call programmed cell death, and it kills itself to avoid tampering with the DNA that is before the telomeres. So telomeres are there to prevent our DNA from being worked on by the enzymes. So as the DNA replicates, it replicates itself. The more replications or the more number of the cell divisions, the shorter the telomeres. It keeps on being cut. They don't code for any proteins, but they just keep on shortening. They protect this DNA from being shortened. Otherwise, it would be DNA that would shorten. But because telomeres are there, at every cell division, it is the telomeres that keep on shortening. And then when finally they are too short because of very many divisions, then the cell undergoes what they call apoptosis, programmed cell death, as you can see in this writing here. So now, cells have an inbuilt mechanism of controlling or preventing cancers, but people have manipulated these. There is an enzyme known as telomerase. Telomerase is the enzyme that is important for this assemblage of these telomeres. So what they do is they go ahead and manipulate. They avoid. They introduce these enzymes in large amounts into their patients so that we keep forming more telomeres and their cells do not undergo senescence. They don't die. But the problem with that it is like a trade-off. You just invite more chances of developing cancers.